Like I told you before, we don't have little chunks of pizza floating around in our cells or other kinds of carbohydrates floating around in our cells. Our cells can't use that kind of stuff. The type of energy that our cells use is called ATP. So what happens is we take the glucose out of the food that we eat. It's going to go through a chemical reaction and ultimately make ATP, which is the kind of energy that our cells can use. The process that does this is called cellular respiration. So it converts the energy from the biomolecules that we eat, in this case it's going to be glucose, into usable energy, which is ATP. And this is what that chemical equation looks like. So as you can see, it's many different molecules because we have multiple different elements together. And this is a balanced equation. It follows the law of conservation of mass. So if we have 6 carbons over here, this 6 applies to the C and the O, so we have 6 carbons over here. We have 12 hydrogens over here. This is 6 is going to apply to the hydrogen and the oxygen. This 2 applies to the hydrogen, so we have 12 hydrogens over here. We have 6 oxygens here. This 6 and the 2 both apply to the oxygen, so we have 12 here plus 6 for a total of 18. Here the 6 applies to the C and the O, the 2 just applies to the O, so there's 12 carbons and another 6 carbons for another 18 carbons. So the number of atoms and types of atoms on this side are equal to the types of atoms that are on this side. What we are doing is we are making the ATP energy from rearranging these molecules. And there are 3 steps in this process: glycolysis, Krebs cycle, and the electron transport chain. If you envision this picture as being a great big cell, cells we know have lots of organelles in them, but this organelle right here is the mitochondria, and you know that the mitochondria is where energy is made. Sometimes we say it's the powerhouse of the cell because energy is made here, and that is because the Krebs cycle and the electron transport chain happen inside the mitochondria. The Krebs cycle is going to make 2 ATP. The electron transport chain is going to make 32 ATP. So there's 34 ATP made in the mitochondria from just one molecule of glucose. So that's steps 2 and 3. Step 1, that is glycolysis, doesn't actually happen in the mitochondria. It happens in the cytoplasm of the cell, and when it's done, the end products of glycolysis are going to move into the mitochondria so we can do this stuff. So let's look at glycolysis. So one of the beginning beginning molecules that we start with here is glucose, and glucose is C6H12O6. This is what a molecule of glucose really looks like. It says we have 6 carbons, 1, 2, 3, 4, 5, 6 carbons. It says we have 12 hydrogens, and you could count all the way up to 12. We would have those all here, and we have 6 oxygens, 1, 2, 3, 4, 5, 6 oxygens. So we are going to take this molecule of glucose and we are going to start breaking it down. So we start off with our reactant over here that is glucose, and it is going to go right here. Now what you'll see is you only see the 6 carbons. What they do in pictures like this is they don't bother with all the extra hydrogens and oxygens. They are just showing you what is kind of important in this reaction, and that's the 6 carbons. So when you see the 6 carbon glucose, what we're really looking at is all of this, but they're only showing us the important parts, which are the 6 carbons right there. So in your notes, they talk about this first part being the energy investment phase. So we have to invest some energy, we have to use some energy to make some energy, which is the energy harvesting phase. So what you'll see here is I have 2 ATP that I am going to use in order to make these ATP down here. So I have to invest some energy in order to make some more energy. So what happens is 2 adenosine triphosphates with 3 phosphates come in, and they each drop off one of their phosphates. So this has 3 phosphates. It's going to drop off one of the phosphates and it's going to hook to my 6 carbon molecule. But because I have 2 of these, I'm also going to have another ATP come in, adenosine triphosphate, and it's going to drop off its phosphate also. So when those molecules leave, they are only a diphosphate, meaning they only have 2 phosphates, adenosine diphosphate, because they each dropped off a phosphate here. So now I have that 6 carbon glucose molecule, Still here, but I have a phosphate on each end, and I actually used up some energy. I had a high energy ATP, and when it left, it was in low energy ADP. So I actually used up some of my ATP here. So I'm literally in the negative in my energy investment phase. So now I have 6 carbons with a phosphate on each side, and an enzyme comes in and cuts the molecule right here. So I have 3 carbons and a phosphate over here, and 3 carbons and a phosphate over here. And remember, an enzyme is going to lower the amount of energy required for a reaction to take place. So that enzyme is going to cut the molecule right here. They don't ever go into a lot of detail about where these phosphates go, but some phosphates come in and they attach to the other sides. So now I have 3 carbons with a phosphate here and a phosphate here, 3 carbons with a phosphate here and a phosphate here. I don't know why they don't ever address where these phosphates come from, but nobody ever does. All right, so now what happens is I'm going to bring in a molecule called NAD. And what happens is it attaches a hydrogen on it. That happens on this side and it happens on this side. The reason that I do that is when I break these phosphates off, I release an electron. And when you release an electron, you need to have somewhere for that electron to go. And this hydrogen is going to capture the electron that is broken off from the phosphate here. I like to think that NADHs are, I used to say taxi cabs for electrons, but now I have to say Ubers for electrons. So this NADH here is carrying a hydrogen that is an Uber for an electron. And over here we have an NADH that is an Uber for an electron. And these NADHs are going to go over to the electron transport chain right here. The NADHs that I made in glycolysis are going to go into the electron transport chain and be useful later. Now, why did I release electrons? So what I had here, I had 3 carbons with a phosphate on each side, and I want to get it down to just 3 carbons. So what I did is I broke off the phosphate here and I broke off the phosphate here. Well, I don't want to just leave those phosphates hanging in out in the middle of nowhere. So what I do is I bring in 2 adenosine diphosphates that only have 2 phosphates. I attach 1 phosphate to one of the ADPs to make it a TP or triphosphate, and I break off this phosphate here and attach it to the other ADP to make it an ATP also. So what I did here is I brought in 2 ADPs, adenosine diphosphates, and they left as adenosine triphosphates. So I got rid of these phosphates and I just have the 3 carbons left. The same thing is happening over here. I break off the phosphates, in comes 2 ADPs, adenosine diphosphates. They grab the 2 phosphates and leave as triphosphates. So I have gained 2 ATP over here and 2 ATP over here. Anytime you break a molecule off, you release an electron. So this NAD became NADH because it is now an electron Uber carrying that H from the electrons that were created here. And what I have left are 2 molecules, and I think your teacher calls it pyruvic acid. It's the same thing as pyruvate, but use the word pyruvic acid because that's what your teacher uses. So what did I form? I formed 4 ATP. I formed 2 ATP here and 2 ATP here. But I had to use 2 ATP up here in my energy investment phase, so I have only gained 2 ATP. I ended up with 2 pyruvates, and this pyruvate is going to go into the next step, which is called the Krebs cycle. And I also gained 2 NADHs, which are really important because they are electron carriers that are going to carry the electrons into the electron transport chain. So this is the picture I like, but your teacher has done the same thing here. So here is your 6-carbon glucose molecule. In comes 2 ATP and drops off the phosphate. So we put phosphates on each side of your glucose here, and it leaves as ADP. Then some enzymes are going to cut it, and so you have 3 carbons and a phosphate, 3 carbons and a phosphate. In comes new phosphates, so you now have 3 carbons and 2 phosphates, 3 carbons and 2 phosphates. Then we break off the phosphates so that these 2 ADP can turn into a TP, diphosphate goes into triphosphate, same thing over here. And when we broke off the phosphate, we released an electron that changes NAD into NADH. These 2 NADHs are going to go into the electron transport chain. So overall, what just happened? All right, so none of this was happening in the mitochondria. All of this was happening in the cytoplasm. We started off with a molecule of glucose that we ate. The process is called glycolysis, and we ended up with pyruvate. Remember, pyruvate are these three carbon molecules that you guys call pyruvic acid. All right, so here's the pyruvic acid. This pyruvic acid is going to go into the mitochondria to start the Krebs cycle. What did I end up making? I made NADH, which are my electron Ubers that are going to go into the electron transport chain in a little bit. And I also made 2 ATP. Remember, I actually made 4, but I used up 2, so I only gained 2. So what is my input? I inputted glucose. I inputted ATP at the very beginning. What is the output? I made pyruvate, which is also pyruvic acid, and 4 ATP for a net gain of 2 ATP. And that's kind of what this is showing you. So the only thing we've really done in our equation so far is the reactants part, the glucose, and we've made a little bit of ATP, but we haven't really done much more with our equation so far. And this kind of says the same thing as right there.